Let's go over the points in chapter 3. Again, this is a lot of stuff, and don't worry, things will start to slow down. Chapter 4 is a little bit quick, and then after that we slow down a little bit. Then the big chapters will be muscle and nerves, of course, but we'll do them a little bit more slowly. So a lot of this, I hope, is review that you've had at some point. So let's get going here. So I started with, and I didn't write this on the slide, hopefully you'll get it, but if you missed class, I wrote on the board cell theory, and I hope you guys are working with my notes. That's why I put them there, so you can actually listen in class and ask questions. Okay, so cell theory, there's 3 ideas. First of all, that all living things are made up of cells and their products. Cells are the smallest unit, having all of the characteristics of life. And if you remember back when we started the semester, there was 8 of those, and you should make sure you go over those for the midterm. And cells are derived from other cells, all right? And then just to introduce the different ideas before we get into the human cells or the eukaryotic cells, we went over some of the key differences between prokaryotic and eukaryotic cells. So eukaryotic essentially means with nucleus, and prokaryotic means before nucleus. And so that brings me to one of the differences, and that's here. There's no defined nucleus in a bacteria. It has DNA, obviously, but it doesn't have a membrane-bound structure protecting it, so it's sort of called the nucleoid. It's sort of collected in a region, whereas eukaryotic cells have a nucleus with a membrane. So the other points where plasma membrane, and often a cell wall, plasma membrane can also have a cell wall. Of course, humans, we don't have cell walls, but other things like plants and fungi, which are eukaryotic, do, all right? Cytoplasm, of course. Bacteria lack most organelles, membrane-bound ones, whereas, of course, we do have those, right? Now here, I wanted to go over the importance of surface area. And you're gonna see, you'll see in my notes too, I started drawing some stuff to try and, explain why cells, or how cells try to increase their surface area versus what you're looking at here with the box. And you're gonna see this particularly when we get into exchange and how different types of cells work, so lung and digestive, all right? But for now, and I'm just referring to my notes, so if you're looking at them on page 1, you'll see my drawing there. So I have this thing about villi and microvilli, so that has to do with increasing the surface area. But why is that? So that is the point here, so going step by step. So if you look at a 1 by 1 by 1 square, or cube, I should say, cube, it has a surface area of 6, right? And that's pretty obvious. And the surface to volume area is 6. Now, obviously, we are not single cells, right? We're made up of many of these teeny tiny cells. So why are they like this? So if you look, if just say it grew 5 by 5 by 5, right? The surface area is going to be 150, and the volume, surface to volume ratio, okay, is 1.2. So that has gone down. Now, whereas if we keep the cell size small, the units the same size, and we just increase the number of them, you can see the surface area now has gone up to 750, and the surface to volume ratio has been maintained at 6. Now, the reason this is important is because for cells to function, there has to be exchange between the outside and the inside. And if there isn't enough surface area to the volume, it's a very inefficient process and the cells will die. So basically what happens is that cells get to a certain point and they have to divide, otherwise they won't function efficiently. All right, so this is the idea here. Okay, and oh yeah, if you remember why I have the egg here. So this is a single cell, right? And once it's fertilized, it very quickly divides into many teeny tiny cells, very, very quickly. So if you ever take embryology, you will learn how that works, or developmental biology. All right. Now the point is here that we have so many different types of cells in the human body. The structures vary, and they vary according to what the cell is doing, okay? But all the structures are essentially still there. A nucleus does what a nucleus does, a mitochondrion does what a mitochondrion does, and so on. All right, there are special things in a lot of these cells, as you'll see, but the essential parts do what they do. All right, now, of course, we use microscopes to study cells because they are small. The point here was comparing the difference in the value, or the not the value, what you get out of these different types of microscopy. And there's more. These are just 3 types. These are the main 3. So we have light microscopy. Use a compound light microscope in the lab. Magnifies about 1,000 times. And the reason it's so important is because it's good for live and dead cells. And you use white light. There are light microscopes that are specialized for different types of light, and use lasers and all kinds of really great stuff. All right. Then we have transmission electron microscopes. This is not using light now, it's using electrons. You can see the magnification goes up tremendously, all right, 100,000. Same thing with the scanning EM. Now these things, you can't study living cells, so this is the problem here. But you can really see surface details with the transmission, with the scanning and the transmission, you can really see the internal structures really well, which you don't get from the light microscope because your structures are still not magnified to the same extent. All right, so together, most of what I'm going to be talking about, these methods of microscopy were used. So I started into the plasma membrane. I went over the idea that this is selectively permeable. All right, we will talk more about that later in the semester in lab. Okay, but the point here is that this membrane is selectively permeable, so it controls what can go in and out of the cell. And a lot of that control comes from the nature of the fact that most of the membrane, or the key component of the membrane, is these phospholipids that we talked about in chapter 2. Okay, so that makes the core of the plasma membrane hydrophobic. Okay, so remember those 2 fatty acid tails in here, and then we have the phosphate head group, which is hydrophilic, and that these things spontaneously organize into this structure. So we have to be able to get things across that membrane, and that's why it is selectively permeable. Now, with that, one of the main ways that we control what goes through that membrane is using proteins. And that's what's shown here. And these proteins are floating around in a sea of these phospholipids, all right? Now, it wasn't at first apparent how these proteins and these lipids were organized, all right? We know now that these proteins can be all the way through, or transmembrane. They can be embedded but not going all the way through, so those are also possible. The other ones are just attached to the outside, as I'll go over. They have adaptations to them, like different sugars, but we didn't know how it was put together. So a lot of microscopy was done to try and figure this out. And one of the key experiments was this one, where they used this technique called freeze fracture. I've never done this, so I don't quite know how it works, but essentially what's done is the cells are frozen and a diamond knife is used to split the membrane apart. And they knew that there were 2 layers, because if they looked using transmission EM, they could see these 2 layers, okay? So they took these things apart, and essentially what they saw now was that the proteins were sort of embedded in this phospholipid membrane. So it used to be thought that there was a sandwich model where there would be like proteins in the middle and lipid on the outside and all kinds of different ideas. But this sort of brought home the idea that these proteins are within the lipid, all right? The other thing was how fluid the membrane was. If you look at transmission EM, I told you that kills the cell, so there's no real way of knowing how fluid this is. So what they would do is start tagging these lipids, and they looked at the motility here. And these things move laterally 10 to the 7 times per second, which is incredibly fast. So that told scientists that this membrane was incredibly fluid, but it really has a difficulty in flip-flopping in this way, which is, as a biochemist, I still have trouble wrapping my head around how this even works, but apparently it does, but very rarely, okay? I'd have to get into some biophysics to try and understand how this is even possible, but apparently it does, all right? But very rare. Now, another experiment that was done to prove that these membranes were fluid and that they could mix, and that these lipids are actually flowing like water, like a think of it like an ocean of lipid, was that they took cells, and it doesn't really matter, human and mouse, it could be 2 human or 2 mouse, it doesn't really matter, but the point is that they labeled one cell with one color and labeled the other cell with another colored dye, and then stuck them together, and they found that the dyes mixed, so the proteins mixed. And this showed, again, that these membranes were incredibly fluid. And I showed this little video in class. If you weren't there, you should have a look at it. It's not like I'm going to ask you to repeat that on an exam, but it just helps you understand how fluid the cell membrane is, okay? And then the third component, so we have 2 components, phospholipids, right, proteins. The third component is cholesterol, and we don't go into this in a great amount of detail in this course, but understand that the reason we have cholesterol in the membrane is twofold, and it's kind of weird. It's like a magic molecule. It does 2 things that seem incredibly opposite. But remember, there's different organisms. We have different viscosity of the membrane depending on how unsaturated these tails are, okay? So remember last chapter, I was talking about how a lot of animal fats are solid at room temperature and fish and plant ones are liquid, which has to do with, when it comes down to the cells that in those 2 types of organisms, likewise, how unsaturated these tails are. But you can tell that it is dependent on temperature, right? So if you leave butter sitting out on the table, it will get softer than it is when it's in the fridge, right? So what the animal has, you can't expect an animal to change its temperature, right? Homeostasis, animals have a certain temperature that they work well at. But what they do is they have cholesterol, animal proteins, or animal membranes, not plant, okay? Cholesterol is an animal thing. I don't think I said that in class. But anyways, understand that the reason that humans and eukaryotes have, and in animals, I should say, animals have these cholesterol molecules in is to moderate this fluidity. So when it's very warm, it sort of restricts the movement of these tails so that it decreases the fluidity. And in cold temperatures, it prevents them from getting so rigid and lined up that they lose their fluidity. So here it's like making it so it's not too fluid. And here, on the opposite side, in cold temperatures, it prevents it from getting too solid. I don't know what I just said, but anyway, so that's the point. Restrains the movement of lipid in warm temperatures and keeps the membrane fluid in cold temperatures. I'm sitting at home and a big truck just went past my window, so I was distracted. So if I said that backwards, just go by what's on the slide, all right? Yes, I'm human, I get distracted. But you see what I'm saying here. Hopefully that makes some sense. And again, the biophysics behind it is, I'm not a biophysicist, but I do understand that this is how it works. And if you look at the structure, it makes sense, all right? Now, with these membranes, like I said, some of them are peripheral, they're bound to the outside of the membrane, and some are integral in that they're penetrating the hydrophobic core. Sometimes they don't go all the way through, but if they do go all the way through, they are transmembrane. Okay, so they go all the way through the transmembrane proteins. And these transmembrane proteins that I sort of focused on for the rest of the class, well, most of the rest of the class, because those are the ones that are going to moderate what can go through the plasma membrane. So with that, transport proteins, and I will go through these again. So some of them are pores and some of them are carriers, some of which need ATP. All right, we'll go over that again. Some of these membrane proteins are enzymes, so they carry out enzymatic reactions that take place close to the cell membrane. Others are signaling molecules, so they carry signals from one side, from the outside to the inside, and there's a change that happens inside the cell in response to that. And others help cells recognize each other. That happens a lot with immunity, for example. Others that we'll talk about when we get into chapter 4, the next chapter, are ones that will join cells together in sheets in different tissues. And others will help the cells attach to proteins on the extracellular side. So things like collagen and fibronectin, right? And that cross-links, it joins then to the cytoskeleton on the inside, all right? Now, let's talk about this selectively permeability, all right? So not everything needs a protein to get through, all right? So keep in mind here, we've got a concentration gradient. So we're seeing high on the outside of the cell and low on the inside of the cell. I'm just doing that figuratively. It could be the other way around. But from high to low concentration, no energy is required in that case. So some molecules, if they are hydrophobic, because remember the membrane is hydrophobic core, if they are hydrophobic, they can just go through the membrane with no problem. And things like steroids can do that, right? So a lot of our hormones are steroid hormones. Remember, steroids are lipids, lipids are hydrophobic. So they can go through. And other things like small molecules like oxygen and CO2 can go through as well. And that go through by the process of simple diffusion, which again, remember, reach back into your memory, is when substances go from high concentration to low concentration, right? Now, other things that are hydrophilic or too big, they need help. And some of that help will come from proteins that make channels. There are pores, that's the other way of thinking of them. They're aqueous, there's a water-filled pore. They're always open, and the only thing that's preventing them from going through is the size, all right? The other ones, sometimes proteins will change shape in response to specific molecules that need to get through the membrane. And if they're going from high to low concentration, no energy is required. These things are not open all the time. They will change shape when whatever it is that enters the protein. So we say these are facilitated transporters, or you'll also see it called carrier mediated. We say that these are passive because no energy is required here. Now, if you're having a facilitated or carrier mediated and you have to, and I'm going to use the term pump, something across the membrane against its concentration, so low to high concentration, energy is going to be needed. And that is called active transport. All right, so if you think again now, and I've put the membrane here because we're talking about diffusion through the membrane, okay? And it's, things can go through very easily if they are small or they are hydrophobic, okay? So you can imagine they'll find their way through, and it might not go as quickly as if there wasn't a membrane here, but it will happen. So it goes from high to low concentration, and please don't write that it goes from left to right. This is just a diagram. High concentration could be on this side, all right? So just remember high to low concentration. All right, and eventually they'll reach equilibrium, which is going to be very important for what we talk about next, which is osmosis. So osmosis is the diffusion of water from high to low concentration across a membrane. And it's important to understand that, again, remember the cell, it's a semi-permeable membrane. Some things can get through and other things can't. Now, water, ironically, actually uses proteins to get through in many cases, in some cases, and in some cases not. And we'll talk about the specifics later on in the course, all right? But suffice to say now, we have water going through by diffusion from high to low concentration. And I know usually we don't think of water this way because it is usually thought of as the solvent, but understand it is a molecule. So if concentrations vary across that membrane, water will undergo diffusion, all right? So in a situation, and they always use the U-tube as an example, if the amount of a solute is the same on both sides, water will be at equilibrium. You won't see any difference here. But if you put on one side, say sugar or salt, okay, what will happen if that solute does not pass through the membrane? The concentration of water will not be equal on both sides, okay? So what will happen is that the water will go from high to low concentration, and it will cause the water level on one side to rise, okay? So and it's because it's going to work until it's at equilibrium. And if you use something like a U-tube, there is a limit to it in terms of gravity, but across the cell, no, okay? Now, this is the phrase I want you to remember: water follows salt, or water follows solute, right? And if you keep that in mind, it's going to make a lot of things make sense as we go through the semester, all right? Okay. This idea of dialysis, I didn't really talk about it in class. We'll do it more in lab. But dialysis applies to other molecules other than water, all right? So when you're specifically talking about water, this is what we call it osmosis, all right? Now, where does this become important? And I think you've seen this before, right? Think of this, and you know, we can't do this in human biology lab, but if you go on to AMP, you will be doing that in week 3, because I'm doing that with my AMP lab this week. But if you have red blood cells, they use this as an example. If you put the red blood cells in a situation where the solute, okay, or the salt concentration is the same on the inside of the cell and the outside of the cell, so we call that physiological saline, that is good, because what will happen is that water will diffuse in and out of the cell at equilibrium, and the cell will be happy, all right? But if you put that cell into a hypotonic solution, so hypo always means less. So in this case, the salt concentration is going to be less, and it's going to be higher inside the cell. So where is the water going to go? Well, it's going to follow the salt, and it's going to go into the cell, and the cell will burst, okay? Especially if that gradient isn't reached before the cell, if that equilibrium isn't reached before the cell bursts. So if you would put, you know, if you would put this in a, say, distilled water, it would eventually burst. Okay. On the other side, hypertonic. So now the salt concentration, hyper is higher, right? Think of hyperactive, it's, you know, more activity, right? More is always hyper. So you can have more salt on the outside here. Now the water is going to leave the cell. We say that the cell is cremated, actually. So this is what happens with human cells. Now we're in the lab, and this is why I bring this up. Okay, I know we're in human biology, but in the lab, we will be looking at plant cells. We don't use real blood in the 105 lab. So we will be looking at this effect using plant cells, which is why I bring it up here. Plant cells are a little bit different because they have a cell wall, all right? We don't have a cell wall, so this is why we have this problem. But as you know, we have skeletons and stuff to keep us rigid or standing upright, but plants do not. So what they rely on is water pressure. So plant cells are actually happier in a hypotonic solution where the water is flowing into the cell. And these cells don't burst because the cell wall is on the outside to prevent that from happening. And that pressure, that water pressure inside the cell pushing against the cell wall, makes the cell what we call turgid, and that's normal for a plant, and that will make the plant happy. It won't be standing, you won't have your wilty plants. In a situation where they're in an isotonic solution, plants will actually wilt because there's no pressure to keep, you know, pushing back on the cell wall to keep the plant upright. So this is when you need to water your plants. Now, when your plant is really, you know, having a problem is when it's in a hypertonic solution. And what you're going to see in lab is that the cell membrane will actually separate from the cell wall. And this situation is called plasmolized, right? And I did not show this in class. It's very corny, but if you're having trouble keeping hypotonic, isotonic, and hypertonic straight, this guy, even though it's corny and it's funny, it works, okay? He uses different examples to help bring this, you know, out for you. So do watch it. It does work. All right. So where does this happen in real life? We're going to talk about this, but I have to go over this idea as well again, so to reinforce what's happening in the human cell, because it's not just water. We also have to think about salt. So let's go over this. So I talked about diffusion, high to low concentration, small molecules, lipid soluble, or hydrophobic. Now, if we have larger molecules, we have channels or pores that are open all the time. And then if we need, these are like gates, you know, facilitated or carrier mediated, the protein changes shape in response to what is being transported, but it's not always open. And on the other side, if it's facilitated or carrier mediated and it's going against its concentration gradient and it needs ATP, we say that it is active transport. Active transport. We don't refer to it as this, even though it is facilitated or carrier mediated. We just call it active transport. And in this case, we need energy to pump these molecules against their gradients. And there's many reasons why we do this in the body. And I'm going to give you these two examples, right? So the main one, the sodium-potassium ATPase, this is the one you've got to get your head wrapped around and why we need it, because it's going to come up over and over again, all right? And I won't always remind you that this is always active. So if you start from the premise that this sodium-potassium ATPase is in all of our cells and it's always active, okay, and you keep that in your mind, and on top of all the other things I'm going to be talking about in the course, it will really help you, okay? So what's happening here, it's a pump. It is active transport. It uses energy, and it's going to pump sodium out of the cell, okay, because the concentration of sodium is higher out of the cell, and it's going to pump potassium into the cell because the concentration of potassium is higher in the cell. So you need energy to do this because both of them are going against the concentration gradients. These are very important reasons why this is maintained, all right? So what happens is that three sodiums will bind to the ATPase. ATP will split, and remember, you're breaking a covalent bond. And remember, the reason that we have these bonds is that we're storing energy. If you break that bond, you're going to release that energy. Okay, so that's one thing that's happening here. The other thing that's happening is that you're putting a large negative charge on the protein, and that's going to affect the shape. Okay, it will affect the shape of the protein. So what's happening now, you've got this negative charge and you have energy input, and what will happen is that will change the shape of this protein. Sodium is going to be released on the other side when the protein changes shape. Now, it is in a different shape. So notice this, they're trying to show you this diagrammatically. This is now open in this new shape here, right? So this now will allow for two potassiums to bind. These two potassiums will bind, the phosphate group is going to fall off. So now the proteins then go back to its original shape, okay? And the potassiums then will be facing now on the inside of the cell, and they will come off. And then we can start again, all right? And this happens over and over and over again. Your body uses a lot of energy to maintain this system. Now, I did not show these two videos in class, all right? I can't remember which one is which, but one of them is very basic. It just pretty much animates this whole thing, and I don't think there's any music or anyone talking. It just shows this in an animation. And if you're very visual, this will help you remember. The other one is also very good, but it's going to give you lots of information that you don't know, but it will explain if you want to know how it works. I'm assuming it's the second one. It really explains how this works and why it works this way, but both of them will animate this so that you can see it, all right? Now, why do we have this? Well, remember, sodium and potassium are small. They're just an atom, so they can easily pass through those pores or those channels that we had talked about. And they'll go down their concentration gradient. So what they're doing is with these pores, and we say that membrane's leaky to these two things, all right? So it's fighting this process, right? Or you could think of this process fighting this process. So, but we need to maintain that gradient in the cell for many reasons. So the gradients must be maintained, and this is why the cell invests a lot of energy to maintain this, and as you'll see, why we need these gradients later on. Now, I'm giving you one example as to, when it comes to osmosis, why we need it. The other reason we need it is for nerve function and muscle function, but this idea here of osmosis applies to all your cells, all right? So you have a situation where, remember, sodium and potassium are part of salts, or they're ions. So they will affect the osmotic gradient on the in and the outside of the cell. And when things are correct, the cell will be happy. Sort of isotonic, the amount of water going in and out will be correct. Now, if for some reason now we end up with an imbalance where the amount of sodium is off, what's going to happen, right, is that if it's higher outside of the cell, the water will go out and the cell will shrink. So remember that we need to try and maintain that balance at the appropriate level. Otherwise, we could go wrong. We could also go wrong the other way, right? And water will flow in and the cells will swell and burst. Okay, so that's another reason we need that sodium-potassium pump to be working and maintaining the proper osmotic gradient. And I use this as an example in terms of exercise. This is something I actually taught in nutrition class. We usually have a chapter on water. But the idea here is the same. So moderate exercise, the body can, work with that. But when you do strenuous and you drink your water too quickly, what will happen is that the salt concentration will drop on the outside of the cell and water will flow in, the cells can burst, okay? On the other hand, if you don't drink enough water, the water will come out of the cell, okay? And the cells will shrink. So that's why it's important to keep your hydration and your electrolyte balances well maintained when you do strenuous exercise. In this slide, I will probably show you one more time this semester. This is the second time, but this is to remind you, the degree of these concentration gradients that are needed on the outside versus the inside of the cell. Now, another pump that we use in the body, and this will come back when we do digestive, is, so just so you know, it's not just the sodium-potassium ATPase. This is also another big one. This is the proton pump, or the hydrogen ion pump. Now, this brings up an idea of secondary active transport, which I want to introduce to you. Okay, so what's happening here is that energy is used to pump hydrogen out of the cell. And the reason that is because it needs to maintain a hydrogen gradient, and that's because protons are used a lot to pump other things against their concentration gradient. So what the cell does is it uses energy to make this gradient. In class, I use the idea of that you should think of it as sort of water flowing over a dam, right? And we can use that water as it flows over the dam to make, because it produces energy, we can make electricity with this, right? So now if you imagine now that this hydrogen is like water and it's now going to flow down its concentration gradient, as you'll see here, right here we have our proton pump, and then it flows down its concentration gradient, that energy can be used to transport other things against their concentration gradient. So something like sucrose, for example, going into the cell. All right, and this then allows for something called secondary active transport. So you're transporting something against its concentration gradient, but you're not using the ATP directly. The ATP was used at the primary active transport here. Okay, hopefully that makes a bit of sense to you. This is why it's secondary. There are also examples of tertiary active transport in the body. I don't talk about them in this course, but when I teach AMP, I talk about that as well, all right? So primary, secondary, and tertiary active transport. But this one here, the direct pump, this would be primary, so active transport. Okay, so hopefully that makes a little bit of sense to you. I just wanted to plant that seed now so that you're ready. Now, please go over these ideas. I did not spend a lot of time on this. I just mentioned it, and I said, please remember. But we have other ways of bringing things into or out of the cell with bulk transport. So, many things in and many things out. So here, endocytosis, what happens there is that actual pieces of the membrane are going to, regions of the membrane are going to invaginate, make a vesicle, and bring in stuff from outside. All right, and this requires energy. I don't talk about it here, but energy is required. I will mention it in a few minutes, one way in which energy is required here. We also have the opposite, exocytosis, vesicles from inside the cell fusing with the plasma membrane and depositing things outside the cell. So it's exocytosis. When it comes to endocytosis, we have a few special types, right? Phagocytosis is when we have the cell membrane actually makes pseudopods and engulf large particles. Macrophages do this, for example. They'll take in a bacterium, all right? Or other things happen as well. Cancer cells are really good at this too. So they make these vesicles, and then this is taken in, all right? We have pinocytosis or pinocytosis. It doesn't matter to me how you say it, tomato, tomato. But here it's just like, we call it cellular drinking. This is usually small molecules that are soluble are brought in this way. So these vesicles that are formed are much smaller than these ones. You can't tell by the scaling here, but usually these are very tiny. And then we have very specific ones where regions of the cell membrane have specific receptors for things on the outside, okay? And these regions are called coated regions. They make a coated pit, and they bring in very specific items from the outside, all right, into something called a coated vesicle. But these are all methods of endocytosis, and they all require energy. Then when it comes to the exocytosis back here, depositing things on the outside, all right? So it's things like hormones, neurotransmitters, we're going to talk about that. But what ends up happening here is that things are made, primarily, say, proteins are made, also lipids, but let's think about this as proteins are made inside of the cell, and say it is a hormone, say it is a neurotransmitter, and it must be deposited outside. So what happens, and we'll go over this, proteins are made in the ER. These types are the ones that are going to be transported out or to the membrane, okay? And they are sent with a vesicle transport system towards the outside. So they're processed in the Golgi, things happen to them, and you get these vesicles that then fuse. Now, if the proteins are transmembrane and they are destined for the cell membrane, when this vesicle fuses, they will stay on the cell surface, okay? But if they are soluble, they are actually found inside the vesicle, and they will be deposited outside of the cell when this vesicle fuses with the cell membrane, okay? Now, the parts of the cell, I'm just going to quickly go over these. You need to try and refresh your memory as to what these are. This, to me, should be review. If it's not, you're going to have to catch up a little bit. So the nucleus, right? So this is where we have our genes or genetic material. The nucleus, as you know, in a eukaryote, which we are, is surrounded by a membrane. It's actually a double membrane system. And the DNA stays in the cell. Some things go in, in the nucleus. Some things have to travel in and out of the nucleus. Proteins, RNA, and the way they do that is through these pores. There is recent evidence that is suggesting that sometimes it doesn't need pores, all right, but that's really new. It's not in your textbook yet. I don't know how well accepted that is yet. But for now, let's say things need to go through these nuclear pores. The nucleolus, remember, this is the place where ribosomal RNA is produced, okay? That's what this nucleolus is, right? We will go over the differences between chromatin and all that kind of stuff later on in the semester. But for the most part, the DNA is as chromatin, and it's bound up with proteins in this region, right inside the nucleus. Now, the nucleus is continuous with the endoplasmic reticulum, or the ER, and I'm going to talk about that in a sec. I like this picture of the nuclear pores. You can actually see that they're very regular. There's a certain amount of proteins that make these pores, and the nucleus can have this all over the nuclear envelope, all right? And the inside of the nucleus, we don't talk about this too much, but there's actually proteins that give the nucleus its defined structure, all right? And that's called the nuclear lamina, all right? Now, ribosomes. We are going to talk more about this later in the semester, but the ribosomes are where proteins are assembled, all right? You get your primary protein structure here at the ribosome. Now, some of these ribosomes are free in the plasma membrane, free in the cytoplasm, okay, in the plasma, or cytosol. And these ones, they're going to make proteins that stay in the cell. These are soluble proteins, and those would be the free ribosomes. Other ones are going to be bound to the ER, and as I was just saying, the ER, or endoplasmic reticulum, is continuous with the nucleus. And those ribosomes are going to make, as we saw with the vesicle transport system, proteins that are going to be released into the extracellular space or going to be destined for the cell membrane, all right? Now, so here you have your ER. So the ones that have these ribosomes on them, where we're making proteins and shipping them to the cell membrane or out of the cell, that's called the rough ER. Now, we also have a flavor of ER that doesn't have ribosomes on it, okay? That's called the smooth ER. Now here, we're not making proteins, but we're making other things, such as lipids, okay? There's also some detoxification that happens there. We make some carbohydrates there. And when we get to muscle, we're going to talk about a type of smooth endoplasmic reticulum where we store calcium, okay? The Golgi apparatus, what happens here is that vesicles from the rough ER move to the Golgi. The Golgi has a sidedness to it. The side that's closer to the nucleus is called the cis face. The side that's further from the nucleus is called the trans face. And what happens here is that as vesicles move from cis to trans, these proteins are modified, all right? They're, sugar molecules are put on there. Things are also sorted for different places in the cell. So the Golgi does a lot of this kind of thing, all right? Then we talked about vesicles. We talked about the ones for transporting to the cell membrane, but there's other vesicles in the cell that don't go to the cell membrane. So they go through the Golgi, and then vesicles come off, and then they stay inside the cell, and they have different functions. And two of the ones that we mentioned in this course are, one, the peroxisomes, okay? The peroxisomes are the ones that help to detoxify things in the cells, right? We will detoxify alcohol here, or toxic wastes. There's enzymes that are called oxidative enzymes. Catalase, if you ever did a catalase lab sometime in your school history, those are found in the peroxisomes. The other ones are the lysosomes. These ones have digestive enzymes that will digest other types of things, all right? So in this case, we're thinking of bacteria that get in here, and then these are going to be digested out. It will also digest out organelles and things in the cell that are no longer functioning or no longer needed. They'll also be digested, okay? And these vesicles stay in the cell, and they will also fuse and get rid of the waste outside the cell, all right? Both of these. All right, the other thing here is the cytoskeleton. We don't talk a lot about it in this course, but it's so important to the cell. There's three types of proteins that make up the cytoskeleton. They each have their own functions, which again, I don't stress in this class, but we have the microfilaments. These are the ones we talk about the most. These are made up of tubulin. These are the ones that help the cells divide. They're also the ones that transport vesicles around the cell, which I'm about to talk about. We also, though, have microtubules. These are made up of a protein called actin. These are really important for cell motility. So cells that move around, so immune cells, like macrophage, they really use the microtubules for that. There's other reasons that we have them, but those microtubules we think about mostly for that, or for attachment to the extracellular matrix. Another one that's not on here, but I will mention when we talk about skin, are the intermediate filaments. And the reason I don't have them mentioned here, not that they don't exist in all the cells, they do, but they can vary between types of cells, what those different intermediate filaments are. We'll talk about one called keratin. That would be an intermediate filament to find in your skin cells. And they're of middle size. That's why they're called intermediate filaments. So the microfilaments are the thinnest, and the microtubules are the thickest, and the intermediate filaments are in between the two in terms of their size and their width, right? Now, in terms of vesicle movement, we talk about this a little bit in this class. Now, these vesicles that I said move to the cell membrane or move around the cell, they don't do this by happenstance. They're actually moving along girders of microtubules. And it's incredible to watch this. I mean, there's videos of this is incredible, and some really wonderful animations. But these vesicles are attached to what we call motor proteins, and they literally walk along the microtubules. And in order to do that, they use energy. There's some really great videos. I wish I had time to just show you these things in class, but you can look them up. But they use ATP, and they walk along these microtubule skeletons, cytoskeleton, and they move and carry the vesicles and move them around the cell, right? So that's one way we use these microtubules, and there's different names for them, whether they're going in or going out, especially when we're talking about neurons. I will mention them when we do neurons, all right? The other place where we see these is with the centroids. The centroid we call the microtubule organizing center. There's two of them. And it's amazing, but they're always at right angles. It's just so incredible how that happens. Anyways, they're they're at right angles to each other, and they organize the microtubules. Like I said, these are what the motor proteins carry the vesicles on. And they're always near the nucleus. When the cell divides, they divide before the cell, right? Because we need these to organize the microtubules for cell division, which we'll talk about later. Now, the incredible thing here is that the way these guys are set up is that they're set up in triplets, and there's 9 triplets set up in a ring for each one. And the microtubules go out from that, and they reorganize from that, all right? And the other place where we see these microtubules are inside flagella and inside cilia, all right? Now, flagella, the only example in the human system where we have flagella is the sperm, all right? So sperm really used for motility, okay? They're long, and as you can see on the sperm here, you get an idea. Now, most of the time when we have in the body, we have cilia. The cilia aren't used for motility for us, maybe in some microorganisms, but really cilia, they beat differently. They're used for moving things across surfaces, and you'll see that when we do respiratory, for example. They move a lot differently. Now, the core of these is microtubules. So the way these things are set up, and I remember the centroids, they had a ring of triplets. And now these ones, and if you look here, this is in a cilium. They are a ring of doublets, okay? Now these things are anchored. They are anchored at the cell membrane, a structure called the basal body. And at this point, you have triplets here, and you have spokes arranged like this. So this is the basal body, and the cilium and flagellum are organized out from that, okay? Now, these things are moving, and the way they move is, again, they use those motor proteins that we were talking about that move vesicles around the cell, okay? And they're attached, they're fixed on one side, and they walk along the neighboring one. So what happens here, all right, so if they were not fixed, okay, if we did not have a basal body, these things would walk past each other, these things would move against each other, which is not what we want. We want them to bend. So what happens is one side is anchored, and as it walks, it will bend the cilium in one direction or the other. All right, so they'll take turns on either side. You can see the full thing here. So this is how we get this sort of movement. And flagella and cilia beat differently. So cilia have this sort of back and forth beating, and the flagellum is more of a whip-like sort of propeller motion. All right, so they beat differently. Okay, but the idea is different. And I actually mentioned the type of motor protein that's involved here. It's called dynin. You're not going to see that on an exam, but just being complete here. All right. Okay. Now, where are we at? Oh, extracellular matrix. So there's a whole slew of proteins on the outside of the cells. They're really important for maintaining structure of tissues and communication and all kinds of great stuff. They are made by the cells, all right, made very often by specific types of cells called fibroblasts, but suffice to say they are sort of a network of proteins on the outside. We have transmembrane proteins that connect them to the cytoskeleton in many cases. These are needed for these 4 ideas: support, adhesion, movement, and regulation, right? And we don't have a chance to get into in detail each of these, but just know that there is this extracellular matrix of proteins, and there's a fluid out here, as you know, and there's a lot of importance that's going on out here as well. Cells do not just exist in fluid and just nothing happens on the outside. There's a lot happening on the outside as well. And this is sort of an example of how some of these are made up. They're made up of proteins and sugar molecules, and they are hydrated, and it's really incredible what's going on out there as well. Okay. Then the last organelle that I use for our segue into making energy or making ATP is the mitochondrion. Now, the mitochondrion is believed evolutionarily to have arisen from bacteria that our cells hijacked. and they are now they divide on their own in the cell. They have their own DNA, which is, you'll see that here, mitochondrial DNA. They have their own ribosomes, they make their own proteins. They really are almost like their own organism inside the cell. And what do we use them for? Because they make most of our ATP, okay? They have a double membrane system. Their inner membrane, it has these folds called cristae. Now, again, think we get to the surface to volume ratio here. If we're increasing the amount of membrane here, we're increasing the surface area, and this increases the ability for these reactions that we need to make ATP, because we've got more membrane in these little structures, all right? So when we talk about this, right, and we use mitochondria to make an ATP, that's their prime function that we consider here. The mitochondrion takes in oxygen, takes in ADP, takes in phosphate, nutrients, and out comes ATP and waste, CO2. Okay, so how does this happen? Your book divides it into 4 steps, and these are a metabolic process. Okay, so metabolic pathways, the point of showing you this, can take many flavors, but understand that there's a series of reactions that take place. We don't go over all of these in this class, all right? But I want you to keep in mind that each step of these metabolic pathways that we use, and we're going to talk about making energy, involves a reaction, and each of those reactions require an enzyme. And as we go through, all right, these things can be linear, these metabolic pathways. They can go through cycles, they can branch, all right? So it's really incredible how all of this just, takes place and is organized, all right? Okay, so when we make energy, our best way of doing this, the most immediate way of doing it, is using sugar. So that's what we'll talk about. Ultimately, we want to make ATP. Why? Because this is our energy currency. We eat food, we capture that energy, and we store it into this last phosphate, the high energy phosphate bond, all right? We make that bond, we're putting in energy, and we're using water, okay? So again, we've got a dehydration synthesis. And when we break that, we're getting off water. And when we break that, we use the water to separate these two, right? And we release the energy, and we use that to, say, drive the sodium-potassium ATPase, or to drive the proton pump, or whatever it is that we're trying to do, have the motor proteins carry the vesicles around the cell, all right? So remember, ATP is recycled, all right? We break the ATP, and we use water to do that. We form the ATP again after we get the ADP, and it goes over and over again. It's a recycled molecule. Now, food, all right? Think about this, all right? You're using oxygen to digest and break down your food. And it is very similar to what happens when you light a fire. You're burning wood, right? And in our case, we're burning sugar. All right. So you know what happens to a marshmallow when it gets too close to the fire, right? It burns up into charcoal right away, and it's mostly sugar. Now, there's a lot of energy that's released there. So instead of doing it in one step, like what happens when you put your marshmallow too close to the campfire, we do this step by step, so we can harness some of that energy and capture it so that we can make ATP. All right. So that's what cellular respiration is all about. This step-by-step process in which we use to capture that energy. So we start off with food, we capture it in different ways. Okay, we're going to have these 4 steps. The prime place is the last one, where we're making most of our ATP, but in order to do that, we capture that energy in high energy electrons that are captured with these electron carriers called NADH or FAD. Okay, so FADH2. So we'll go over that right now. Okay, so we have 4 steps. And for this class, the first step is glycolysis. This is where you're splitting sugar. So glyco, sugar, lysis, splitting. And you split this 6-carbon molecule into 2 3-carbon molecules called pyruvate. Now we make a little bit of energy here. We call it substrate level because we're doing it right from the actual substrate, which is sugar, all right? And this, importantly, it takes place in the cytosol. It does not require oxygen. So if you're working out and you're not oxygenating your tissues very well, you're still making some ATP without the oxygen, right? Very little, but we'll talk about this later on when we do muscle. But most of our ATP is made in the mitochondria, but now we have to get this pyruvate molecule into the mitochondrion, and that happens in the preparatory step. Very often you don't see this listed as this, but your particular book calls this the preparatory step. So it moves the pyruvate into the mitochondrion, loses a carbon. Now remember, if you're losing a carbon, you're breaking a bond, you're making energy, so releasing energy, and I'll go over that in a sec. And then you have a 2-carbon molecule that gets into this metabolic pathway called the citric acid cycle or the Kreb cycle. And here you're making a little bit more ATP, but you're making a lot of energy carriers. So remember, there's electrons are what's going to harness that energy from the breakdown of that sugar and put it in a situation where we can now harness it to make ATP. So you're not making a lot of ATP here, you're making some, but not very much. But you're making a lot of these guys, all right? Now, where we make most of our ATP? In the last step, which is divided into 2 parts. One is called electron transport, and the other one is ATP synthase, or chemiosmosis, all right? So electron transport is going to set up a gradient that we use in the final step to make ATP. So I will go through that with you quickly. All right, glycolysis. So here, the key thing here is we're starting with our glucose molecule. And remember back to that idea that I gave you with that car. You know, in order to start a reaction, you need to put in a little bit of energy. So that's what's happening here. Now, in order to make ATP, the cell invests 2 ATP molecules per glucose to get the process going, all right? Now, then we have a series of reactions, all right? And what's happening then is that as we go through here, we're going to make 2 ATPs, but you're going to make some of these electron carriers, and you're actually making more ATP in the long run with these electron carriers, which you'll see shortly, okay? And what's happening now is that you're making pyruvate and a bit of waste, water, okay? You're going to pee that out, all right? So let me just move forward in my notes so that I'm following, making sure I'm telling you what I'm telling you in my notes. Okay, so energy investment, a little bit of energy yielded, all right? So you take your 6-carbon sugar and you get 2 3-carbon molecules of pyruvate, all right? Now, now we have to get that pyruvate into the mitochondria. So this would be your intermediate stage here. What do we call it in this textbook? Preparatory step. Yeah, it's kind of a weird thing. I've never encountered that before. Preparatory step. Now, notice what's happening here, all right? You're taking a 3-carbon molecule, you're ending up with a 2-carbon molecule, you're making some waste, CO2. Now, you've broken a bond here. Remember, inside each bond is some energy, and the cell is not going to waste it. It's going to capture that energy and make another electron carrier. All right, it's going to, we'll add that electron to your electron carrier, so you make a molecule of NADH for each of those pyruvates. All right, so you're not wasting that, but you need the 2-carbon molecule to start you into Kreb cycle, or the citric acid cycle. And this cycle, it goes through, it makes some ATP. All right, you're breaking bonds. The energy is captured again using electron carriers. So you're going to make 3 molecules of NADH and 1 molecule of the other electron carrier, FADH2, for each time the cycle goes through. So you do this twice for every sugar molecule. All right, so you're making a little bit of ATP, but still not a lot. Now, the prime function here is to make these electron carriers. All right, and I just showed you some of the reactions here. You don't need to memorize these for this course. All right, I just want you to see, if you follow through, you can see that each step, things happen and energy is being captured, all right? Now, this leads you to the last step, which, like I said, is made into 2 parts. The first part is the electron transport chain. Now, what's happening now is now you're setting yourself up to make your ATP. And the way the cell does that is, again, not directly from the electrons, but setting up a gradient of protons, all right, so of hydrogen molecules. So what happens is that these energy carriers in the mitochondrion All right, so you're inside the mitochondria. This is the mitochondrial space, and this is in between the two membranes. So this is the cristae on this side. Out here, it's not shown, but it would be the outer region of the mitochondrion. And these electrons are passed like a hot potato down this thing called an electron transport chain. There's a series of proteins here. Now, what's happening as the electrons are passed is they're releasing a little bit of energy. And that little bit of energy isn't used directly to make ATP, but what it is used for is to pump hydrogen molecules into the intramembrane space. So we get a gradient of hydrogen, okay? What is stimulating that hot potato toss? Well, what's stimulating that is oxygen. Oxygen, if you think about it, is an oxidizer. It grabs electrons, all right? Try to think of that from your chemistry. But suffice to say, what is the point of all that oxygen they're taking in when we're breathing? Well, we need it to make ATP. And the reason we do is because it pulls the electrons down at the electron transport chain and accepts them. And when it does so, it makes water, which we pee out as a waste product, all right? So this is one of our waste products. I already showed you a few places where we made CO2, which is our other waste product from metabolism, all right? So here we are. The function of oxygen is to pull those electrons down the electron transport chain. Now, what happens when we have that gradient of hydrogen? So what happens then is it goes through the last step, the ATP synthase. And this gradient, now remember, this is like the water flowing over the dam again. The water's flowing over the dam, and what's happening is that energy that is released as this is falling over the dam or through this synthase, that energy is now going to be captured to make ATP. Okay, so I hope that makes some sense, all right? And this is how the cell does it, right? So if you go back, and I think I have it on page 15. So you take in, look at the reaction there. You take in sugar and oxygen, right? So we breathe in the oxygen. We give off CO2 and water as our waste, right? So CO2, every time you take off a carbon, it's going to be released as CO2. That's your waste. And that water is generated as well, right? So primarily here at the end of this electron transport chain, because oxygen is pulling those electrons down, and when it does so, it will grab hydrogen, it won't make water, okay? All right, make sure I didn't skip anything. Okay, so if we look at the accounting, right, you can see most of our ATP is made here at the last step, oxidative phosphorylation. So electron transport and ATP synthase. The idea here is about 36 ATPs per glucose molecule. We lose some energy in moving things around and pumping that ATP out. So we lose a bit. It's about that. And these two videos, again, these are really great. They animate the electron transport chain and the last step, the ATP synthase. All right, so really great to watch. It animates the whole thing, and hopefully it helps it make sense. The point of this last slide is just to show you that the reason we take in all these other foods is because, again, we can use those to make ATP. They just enter this process in different places. Okay, so here we're just talking about sugars. But remember, fats actually have more energy available in those bonds than sugar does. So obviously we can use it to make ATP as well. It comes into this process in a different way. Even proteins can be used to make ATP, and they enter in different places in the metabolic pathways that are involved here. Okay, so hopefully that helps. I know this again is a huge chapter, but hopefully a lot of it is review, and I've refreshed some of your memory on this, okay? Okay, so let us stop the video, and thank you very much.